I want to give a really quick explanation of segmentation in vertebrates. I talked about it in Drosophila. In vertebrates, it can be a little bit confusing, but I just want you to see another way to do segmentation. So we're going to start out here where we're looking at the formation of the neural tube and somites. The somites are the blocks of mesodermal segmented tissue. So somites are mesodermal and they're blocks of tissue. And so you can see these somites here. So each of these blocks of tissues is a somite. They're paired. So a pair of somites like this is going to be one segment, and in the middle we see the notochord. So here's the notochord running down the middle of these, and this I think is just in a zebrafish. So the somite formation, it begins in the anterior portion of the trunk. These things called paraxial mesoderm. Paraxial mesoderm is like just like a big chunk of semi-undifferentiated mesoderm. So it's already gotten to be mesoderm. It's paraxial. It's like on the sides of the notochord. And now it's going to get organized into these somitomeres, like down here. So they're kind of like lumps. And then they get compacted and bound together by an epithelium to form the somite. So you basically have this like actual epithelial boundary around these blocks of cells here. And what that means is that they can undergo development relatively isolated from each other. So signals from this somite here don't necessarily affect this signal here. It's only really specific signals that are going to get to these, which is kind of a neat way to do this. The number of somites that are in an organism, that usually tells you how far development has proceeded. So I used to do zebrafish just really briefly. I did a quick project on zebrafish in grad school, and the way that you would tell how old the zebrafish was by literally counting the little somites down in its tiny little body, and that would tell you how old it was. So this is just looking at a little bit of segmentation and somite formation right here. So here's kind of where we're looking. So here we see these somites that have formed, and they're gonna be working their way from the tail end of the embryo, and they're gonna get pushed up. So you make the anterior ones first, and they get pushed progressively more anterior by the formation of the posterior one. So they're getting pushed forward in a line. So here we've got our developing neural tube, and then here's our notochord running down the middle, and then our paraxial mesoderm is gonna, some of it will become presemitic mesoderm, and that's gonna be found on either side of this notochord. Okay, so if we do a cutaway here where we already have these somites formed, here's what we'll see. We'll see our neural tube, we'll see our notochord underneath the neural tube, just like we saw before. And then on either side of this, we see the paraxial mesoderm. In this case, this is going to be the semitic, the somites forming right here. Okay, if we go further out, we see these things called intermediate mesoderm and then lateral mesoderm. We might have time to go into these more later on in the class. All right, so the mesoderm invertebrates, these are the different regions of it. So we have the lateral plate mesoderm. This is like circulatory system, lining of body cavities, and then the limb components, not counting muscle. The paraxial mesoderm, that's going to become the somites. So what do the somites become? They're going to be the framework for the organization of the axial skeleton. So the vertebral column and the ribs. And the dermis and the muscles of the back as well. They're also going to become the skeletal muscles of the body wall and limbs. So they're forming a lot of these segmentally repeated structures, mesodermal ones. So how do you form this? So how do you start with sort of like this big chunk of tissue in the back and then have like little bits bud off of this? And this is sort of like a funky like math/physics problem. And so work by Olivier Porquier figured out that this gene called Hairy was oscillating in its expression patterns just in time with somite formation. So he would see it in the presemitic mesoderm. Here down the middle is our notochord. All of this stuff that is like in this U shape, this is all the presemitic mesoderm. In the presemitic mesoderm, he would see it kind of like as this big, broad expression pattern. And then just before a somite was about to bud off, it would resolve itself at the anterior part of the presemitic mesoderm into a stripe. Okay, and this stripe would eventually be present in the posterior half of the newly budded somite. So we see this somite budding happening exactly in concert with the oscillations of Hairy in the presemitic mesoderm. Hairy, which in vertebrates is called HES or HER, is a target of the Notch signaling pathway. And so again, these are expressed in this dynamic pattern. They cycle in the pre-Semitic mesoderm, and they cycle exactly in timing with the generation of the somites. And so this is just looking at the actual expression patterns. And so all of this back here is pre-Semitic mesoderm. And you can see that just before a somite's about to bud off, this HES mRNA resolves itself into a little tiny stripe. And so here we have it in the here we have it in the back half of the newest formed somite. And this is just looking at sort of like this like wave of stripy expression that's happening. And so you get the wave of expression from the pre-Semitic mesoderm resolving into these stripes in the posterior half of each somite. So just a reminder of Notch signaling. So here we have a Notch in the cell membrane, and in a next door cell, we have a DSL ligand, and these include delta. I think it's delta, seri, and lin12. Gosh, all the lins get me all confused. I think it's lin12. Who knows? It could be anything. I don't have it in my notes. So here we have delta. That's my personal favorite one, and it's also the one that rules the show in somatogenesis. So this cell has a delta ligand. This cell has a Notch receptor. And so these chitty-chat with each other. And when they bind to each other, that causes this Notch protein to undergo intramembrane proteolysis. So inside the cell membrane, it gets like chopped in half. And the inside cell half translocates to the nucleus. So it doesn't bother with second messenger systems. It translocates to the nucleus, and it works together with other proteins in the nucleus to activate gene expression. So it's a co-transcription factor. It itself does not bind to the DNA. It binds to transcription factors that can then express these downstream genes like HES. So here we're just looking at this, how it works as an oscillator. And it's kind of a neat oscillator. It uses the lateral inhibition property of Notch signaling pathway to work. So I told you before that basically Notch signaling has this lateral inhibition property where you basically can have one on cell surrounded by a bunch of off cells. And this is sort of how this works here. So let's look at these two next door neighbor cells. So they both start out expressing both Notch and delta. And so they both kind of have like this same pattern coming out of it, where you have a Notch being activated by delta, traveling to the nucleus, turning on HES. In this case, it's called HER for... Do you want to know? You do want to know. So HES is for hairy enhancer of split. Those are the two Drosophila genes that are homologous to this like whole suite of vertebrate genes. And HER is hairy enhancer split related. It's like a mouthful, but they're all homologs of each other. They're all like have a lot of the same tasks in a cell. All right, so Notch activates the HES genes, and then this gets made into protein. And this protein, HES protein, is actually able to repress the formation of delta. So you stop putting delta into the cell membrane. If you have delta on, delta is going to be in the cell membrane and be able to activate Notch next door. So that's how you turn off HES in one cell and keep it on in the other, right? So it's going to be kind of like random which of these cells happens to have faster Notch signaling to turn off delta in it. So no delta in here means no activation of Notch over here. Okay, but that only gives us like an on-off pattern. What about the oscillation pattern? Well, the oscillation pattern comes about because the HES protein actually represses its own transcription. So if you have HES protein around, it's a negative feedback loop. It turns itself off. So you basically get it turning on, and that's just going to have to do with like the physics of how fast this happens, how fast it gets transcribed, how fast the signal gets into the nucleus. So it gets turned on briefly. You get a lot of this HES protein, turns off delta, so no Notch signaling in this cell. So this cell makes nothing at all. And then it turns itself off, and then you can have it happen in another round again. Okay, so that's the oscillating part of it. So we have this on-off part of it that gives us a stripe, and then we have this oscillating part of it that is in time with somite formation. And so this is looking at the pre-Semitic mesoderm of an embryo that has its HES protein tagged with luciferase. So that means you can like visualize it. If you stick on luciferin, it'll glow, it'll emit light. It's so neat. This is like old-fashioned-y. You can see this is from 2006. Nowadays, we usually just tag it with like GFP or something. All right, so this is looking over time, is kind of down this way and then across this way. So time like this, and you can kind of follow the oscillation. And so the researchers, of course, followed the oscillation for us, so we don't have to like build a movie inside of our heads. And they looked at 2 spots, so spot 1 and then spot 2. And so spot 1 on here is in red, and spot 2 is in black. So spot 2 is going to be where we expect to see the stripes starting to really resolve themselves. And this is exactly what they see. They saw oscillation in both of these 2 regions, and they were slightly offset from each other. That's telling us that there's this wave of expression that's traveling through the presemitic mesoderm. And that's exactly what we see here. So we see it dark here. Dark here, it starts to go away here as HES is turning itself off. And as it's going away over here, it's getting darker up in the anterior regions. And so it's really like traveling like a wave through the presemitic mesoderm, resolving into a stripe of HES. And we think HES, like, is it just a universal oscillator? And if we want to think about it in terms of a universal oscillator, the really important things to think about are these like delay steps. So we activate HES, and then there's going to be a delay in making mRNA, and then a delay in how long it takes to make protein, and then this protein can go and repress the HES. And so thinking about these delay steps, these 2, as well as the delay steps of how long does it take to break down this mRNA, how long does it take to break down the existing protein, that's going to give us the timing of the oscillation. And so that's what this research was looking at here. This was looking at the oscillation of HES mRNA production. So that's the HES expression pattern, which we know it's oscillating with protein synthesis at a normal rate. And then they looked to see what would happen if they attenuated the protein synthesis. So if you attenuate the protein synthesis, you of course get like sort of these like lower amplitude curves. And then here they have it with protein synthesis at a normal rate. And then they allow for noise in the system. Now, what's really important to see here is that with normal rate protein synthesis, or even low rate protein synthesis, and even allowing for noise, you still get these really distinct peaks here. And so that means that even if you kind of like bump the system a little bit and the system's noisy, you're still going to get this acting as an oscillator. And so the big question now is this acting as an oscillator in other kinds of developmental contexts? And the answer is like probably yeah. So that is a quick look at somitogenesis, and then kind of our first look at a little protein and RNA machine that can be used to pattern tissue in a repeating fashion.